Hello students, welcome to Botany class. And today we are seeing about the growth hormone. That is a continuation of the yesterday's class. Okay, so what are growth hormones and how they will affect in the plant body? Growth hormones are the ones which are present in the plant body and they will work along with the light intensity. Okay, so whenever the light intensity is more, then they will, wherever the light intensity is there, then they will go there and there does their action. Those are the hormones, plant hormones. So these are called as the phytohormones. And these phytohormones can be mobilized very easily from one part to another part. And all these growth hormones in the plant body are called as the PGR, plant growth regulators. Okay, so in the competitive exam also, they will ask us with the word as the PGR. And these PGR will be, these hormones will be present in a small quantity, and then they will do some specific work in it, and they will show the much more plant growth of the specific part where they are present. Okay, those are the plant growth regulators. And these plant growth regulators are of two types. That is the one is the growth inhibitors and the another one is the growth promoters. Growth inhibitors means they will control the plant growth in the form of the seed dormancy and the early leaf fall and the fruit fall. Flower is going to be disturbed. That is the flower fall can be seen. The bud cannot be, seeds cannot be starts their germination like that. So that is called as the inhibitors. So those are two inhibitors, abscisic acid, ABA, and ethylene. And the growth promoters, that means they will show the plant growth at any cost in different manners. Those are the plant growth promoters. And these promoters, growth promoters are the auxins, gibberellins, and the cytokines. Now comes with the auxins. Auxins are the first and the most important plant growth hormone, which is increasing their growth with the cell elongation. Internodal length is going to be increased and the stem growth will be much more. They are stored in the stem tip and they are produced, otherwise synthesized in the root tip. So they will be traveled through the xylem vessels in the upward direction, and only in the upward direction the traveling of the auxins can be seen. Okay, from the synthesized to the stored plant part. And these will be first of all observed by the Charles Darwin and his son Francis Darwin. Okay, both worked on the coleoptile of the canary grass. Coleoptile means stem tip. So there they have given some hormones are present which are showing the growth promotion. And then the Wendt came, that is a scientist name, and he has given how these auxins will work out with the light intensity. And he has observed this with the Evina curvature test, what we'll say. And these are, again, he has done his experiments in the agar jelly. Okay, agar jelly. Next comes with and these will show the apical dominance. Apical dominance means they will control the lateral bud domination, lateral bud formation, and any other hormone that is present in the plant body. All are suppressed by the auxins, and they will show only one function, that is the apical dominance. The stem tip is going to be elongated. That is the apical dominance, what we can say. And they in the tissue culture lab, in the tissue culture and the stem cuttings, there if we supply the auxins in a limited number, then they will enhance the fruit formation and the shoot formation with the different ratios. If the small amount of the auxins are supplied, then the shoot formation can be seen. And a large amount of the auxins are applied for the stem cuttings, then the root formation will be seen in the absence of the root for the stem cuttings. So like that, we can enhance the root formation. And the artificially synthesized auxins are there. Those are the 2,4-D and 2,4,5-D. And these two will be acting as the weedicides. That means they are removing the dicot weeds in the monocot agricultural plants. And these are not at all doing any harmful effects to the agricultural monocot developed plants. And they will delay the abscission, that means the early leaf fall and the fruit fall in the plant body. And they delay the senescence also. That is the death of the cells can be seen. And this they will enhances the parthenocarpy and they will do the sex determination. That means the female flower productions will be increased than the male flower production by a supply of the auxins in the plant body. Example, Cucurbitaceae family members. And the next one is the gibberellins. In the gibberellins are the one of the hormone which will be somewhat acidic in nature, that is weak acidic in nature, and these are called as the Gibberellic acids, and these are synthesized, otherwise identified more than 100 in number. So each and every hormone is given the number as the GA1, GA2, GA3 like that only. And this was first discovered by Kurosawa and rice plants, which are causing the infection with the fungal organism called as the Gibberella fusicori, where the gibberellic acids are produced more by this fungus into the plant body, and the plant got the Botany disease in them. Okay, and these will be synthesized in the apical parts of the stem and the young leaves and stored in the matured leaves and the root tips. Okay, in the roots they will be stored, and this passage of this gibberellins will be done with the phloem, downward direction. Okay, and this in this one GA3 is going to be used in a large number in the agricultural field area. We will say this as the, it will increases the seed germination in the lettuce plants in the absence of sunlight also. Okay, and it removes the seed dormancy in the monocot plants, and it removes the bolting. It increases the bolting also, we can say, that is, it removes the internodal distance. It decreases the internodal distance between the in the stem, and it increases the elongation of the internodal length. So that is called as the bolting process. This can be seen in the cabbage, where the internodal length is very less. So there will be forming, the leaves will be forming as a holes. That is the removing of the genetic dwarfism also, we can say, in the gibberellins. And next comes the cytokines. Cytokines are the hormones, growth promoting hormones, which are basic in nature. And this was first discovered by Skoog. And these are found in the root tips and the stem shoot buds. And these are stored in the matured and young plant parts, that is the young leaves and buds, lateral buds will be having this cytokines, and these will be increasing the, just like the similar action as per the auxins, and along with auxins, so we can use the cytokines in the tissue culture lab for showing the root promotion and the shoot promotion in the stem cuttings and in the tissues. So they will increases the cell division and increases the cell elongation, and they will control the apical dominance that was caused by the auxins, and they will be, and so with that the branch formation will be more with the help of the cytokines. That's the one what we can say over here. And these will be seen in the used in the agricultural, in the tissue culture lab in the 1:3 ratio. That is, auxins are 1 and cytokines are 3. Then the root promotion will be seen. When the cytokines are 3, 1 and the 3 auxins are 3 and cytokines are 1, then the shoot promotion can be seen. Like that, we will see the auxins and the cytokines ratio in the biotech labs for increasing the root and the stem. For the stem cuttings and the tissues, organelle formation can be increased by the cytokines. Next comes the seed, the hormones which controls the growth. Those are the growth inhibitors. The first one is the ethylene. In this 5 hormones, auxins, gibberellins, cytokines, ethylene, and the abscisic acid. Ethylene is the only one hormone present in the gaseous state. And this occurs, this will be occurring mostly produced in the ripened fruits and in the matured tissues and the leaves, and stored in the young buds and the young leaves. Okay, and this, and in the root tips also, in the roots also, this can be stored, and this will be undergoing, and it will be increasing the seed dormancy in some flowers and in some plants, and decreasing the seed dormancy in some plants, and it overcomes, that is the, it takes up the seed dormancy and bud dormancy in some plants, and it increases the vase life and the shelf life. Vase life means With the flowers which were taken out from the parent plant, the life will be increased in them by the sprinkling of the ethylene, and the fruit can be matured by sprinkling of the ethylene, okay, in the godowns. And the shelf life, that means the leafy vegetables can be stored without any damage of the leaves in the godowns, the cold storages, by sprinkling the ethylene. That is the plus point in the ethylene itself. And the next one is the abscisic acid. Abscisic acid is also called as a stress hormone, which is a growth inhibitor, and it increases the closing of the stomatal apparatus, and it increases the bud dormancy and the seed dormancy in the monocot as well as in the dicot plants, and it increases the abscission also. Okay, and it does not allow the parthenocarpy, abscisic acid. And with this can be combined with the first topic, first chapter, transportation in the plants. Okay, so if you are having any doubts, you can message me and prepare well with this topic, because we are expecting a session in reason from this topic. Okay, all the best, and please subscribe this channel. Right, thank you.